Welcome to this Labroots podcast, The Power of Attraction, exploring Dynabeads in sample preparation. This podcast is brought to you by Thermo Fisher Scientific. Partner with our global team of experts to navigate and conquer challenges from discovery to commercialization with fast, reliable, and cost-effective solutions. To find out more, go to thermofisher.com/Dynabeads. Now here is your host for this podcast, Senior Product Manager of Dynabeads Products, Claire Mount Lund.
So welcome listeners to the first in this series of podcasts, which is entitled The Power of Attraction, Exploring Dynabeads in Sample Preparation, your window into the world of scientific research. Before we go any further, I just wanted to make a note that all of the products and the techniques that we'll mention in this podcast series are for research use only and not for diagnostic procedures. And with that, we can jump right into the podcast. So today's episode is about Dynabeads products, and we are joined by one of our experts, Erling from Thermo Fisher Scientific, world leader in serving science. So today we're going to be exploring really the innovation and the innovative role behind Dynabeads magnetic beads. And with that, before we jump into exploring Dynabeads in a bit more detail, it'd be great if you could just introduce yourself a little bit, Erling.
Yeah, thank you. It's great to be invited. My name is Erling Finne. I'm a senior staff scientist here at Thermo Fisher's scientific site in Oslo, Norway. And I've been working with magnetic beads in some sort of another since the start of this venture in Norway.
Sounds like there's an intriguing journey that we can go on in this podcast to find out a little bit more about that background. So maybe, I know Dynabeads is a technology that's been around for many decades, actually has a long history. Maybe you could take us actually right back to the origin story of where everything began.
Yeah, it's quite fascinating because you can actually trace the use of this Dynabeads technology back to space. We had a professor at the Technical University in Trondheim, Norway, who was working with polymer chemistry and was attending a conference where NASA claimed that to make completely round, equally sized, mono-sized polystyrene particles, you would need to be in space, away from the natural gravity on Earth. And this Professor Ugelstad, who we have with us today, he took on this challenge using his experience from thermodynamics and competence to say that I want to make this on Earth in our laboratory here at the university.
Incredible. Wow, what a man.
He was, yeah.
Yeah. And it sounds like he has a interesting background as well. Maybe you could tell us a little bit more about Professor Ugelstad so we can get to know him a bit better. Yeah,
that would be a pleasure. I was one of the fortunate who had Professor Ugelstad as a professor. So I know him from that side, and also later when Dynabeads and magnetic particles became a commercial product, he was very much involved in all the details around the first commercial products. Even though he did not join the company as such, he was always there and showed his great interest. And I think that is one of the unique properties with Ugelstad, that he had an almost childlike curiosity and playfulness, as well as being an absent-minded professor with a lot of funny stories. So he was a great inspiration for us students, and also in the beginning of the commercial venture of Dynabeads and magnetic particles.
Sounds definitely like an interesting man to know, and it's not everyone that would take on a challenge from NASA like that either. So very interesting.
Yeah.
But so that was, of course, we know Dynabeads has been a very good success and a great innovation that came out from Professor Ugelstad's team. were there any other innovations that he came up with? It sounds like he had a lot of curiosity out there. I heard a rumor around some military buttons, for example.
Yeah, that was one of the stories he liked to tell, when he had an idea to make use of some natural polymers that were left over from the fish industry, to actually use this to make cheaper and good buttons for military uniforms. And that seemed to go well in all their testing, etc. But once these uniforms were subjected to the washing machines and the detergents used by the army, they all disappeared. So the customer was not very happy, and maybe that's one of the lessons, to really understand your customers before launching and going into too much product development.
Definitely good to learn from those who came before us, and also good to see that the path to innovation isn't always a straight line. Sure. There can be failures and deviations along the way. So interesting. The Dynabeads, that has definitely been a success. Maybe you could take us a little bit into that history again. So he identified these particles.
Yes, so he designed a swelling process, which is used called the Ugelstad process, where you can go from a tiny seed particle and up to any predetermined size of mono-sized, equally sized beads in any range from 1 micron and up to maybe 100 micron. So he was able to do that for non-magnetic particles, as we used to call it, so the step before we make the Dynabeads. And those products became very important in the chromatography industry, creating beads with very low back pressure in columns, because they're all equal sizes and will pack in perfect packing. But Ugelstad also, and at that time, realized that if you were able to magnetize these mono-sized particles, it would be a very valuable tool to isolate material from all sorts of samples, maybe also dirty samples where you have other solid material present. You can use the magnetism to pull the stuff you want to save out of the mixture. Mm-hmm. So the initial making polymerized beads was patented in 1976, and by was it 76, around there? But around 86, where he was also able to patent the way of making these super paramagnetic. That is, that once you have them in a magnetic field, they will go to the magnet. You remove the magnetic field, they will lose all their magnetism. So you don't create clumps of beads. They behave just as any particle when there is no magnetic field.
And then how did we get to the point, so we got the magnetic beads? How did he or his team find out how we should apply these products? Did they know from the start? Was there an aim, or did that come later?
That certainly came later, and also since Ugelstad and his team at the university, they were polymer chemists. So, but with his enormous skill of catching or getting into contact with people and traveling around to different universities, he was able to create a lot of interest for the use of this, and a lot of ideas came up. How could this technology be used to isolate cells? Could it be used to pick out cancer cells from bone marrow and cure patients? Could it be used to isolate small molecules? So at this time, there was an explosion in different kinds of research and new applications coming up. And we also saw that this came at about the same time as the hybridoma technology for developing antibodies and large-scale production of monoclonal antibodies was really maturing. So combining these two, that you have solid particles that can go into any type of material, and an antibody that can capture a cell or a molecule or whatever, that really made it a powerful tool that was developed into a lot of new applications.
Sounds like an incredible first step, and also another role model moment for us from Ugelstad of making sure we collaborate to get the best out of all of our technology as well. So really nice story to hear about. So then, since then, yeah, tell us a little bit more about what's happened with Dynabeads since then. You mentioned the antibodies. What other kind of applications have come from these?
Yeah, so this first idea of isolating cells and working with cancer research is certainly something that's still really important, and now also accelerating. The near technology to that, it also immunoassays, where it was discovered that instead of running immunoassays in plates where you have antibody only on the side of a small well, you have the sample in between and not very good contact between the sample and the side of the well. With the beads, you can have the beads present throughout the sample and get much quicker isolation, much better yield. So that has also been very important for new technologies within protein separation and protein analysis. And all of these procedures can also be automated based on the magnetic separation technology.
Yeah, sounds like it's definitely taken us some steps forward by having access to technology like Dynabeads, and it's having impact, I know, around the world. But how about for Norway? Has it made any impact here?
Yeah, so actually this is one of the great success stories for biotech industry in Norway. It has been recognized as one of the major inventions in Norway, and we're also certainly been a driver for developing biotech industry, creating spin-off people leaving the company, creating a much better infrastructure for biotech industry. So that has been really important for Norway as such. Yes.
Incredible legacy for Professor Ugelstad and Dynabeads.
Yeah, so I mean, he lived to see a lot of it and was, of course, very proud and always involved. But
technology is definitely living on. So incredible. So can you tell us a little bit about what makes these Dynabeads quite so unique and versatile in the way that they can be used?
So one of our slogans is that you can capture anything with Dynabeads, because the main feature is then that the combination of size, how we make the surface coating, and I would say also that this is a very reproducible and robust production process. So we can design beads depending on the application. The surface can be modified to tailor to the application. You can have chemistry that is on the surface that is specific for the application, or you can have a biological derivatization of the surface that's needed for your application. So building on the few building blocks that we have as the base particles, we can branch out into a huge variety of different applications.
Interesting. Definitely, yeah, exciting how many applications. It seems almost endless possibilities for these Dynabeads. What about how, like, some of the key features of the Dynabeads and how they kind of interact with the targets or the magnetism of the products? Any features you'd like to highlight there? Yeah,
so when you have the beads in a tube, it looks like a brown soup. And then the principle is, of course, that once you place this in a magnetic field, where we have magnets on both sides of the tube, especially if you have a lighter background, you would see immediately that the beads go to the side and you can decant or remove the liquid. So it's a very easy process of separating what you want to have on the beads and throw away the remaining. The key property here is, of course, that you don't capture and throw away anything of your valuable target.
Mm-hmm.
So the very important feature that we are working with is to create these surfaces in such a way that there is low unwanted binding, low non-specific binding to these beads, but only that we get the specific isolated molecule.
And yeah, you started to show us a little bit here. Can you take us through, like, step by step the
Yes.
process of, like, say I came to you and I have a blood sample, an imaginary blood sample. What would my basic procedure be?
So all our basic procedures are based on three steps, maybe. That you first step, you capture whatever you want,
And
then you remove everything else, and then you do some analysis on your isolated target. So the first step, you have beads that have either a chemistry or a biological function that can act as a fish hook for your sample. You remove this, and if needed, you do the washing to remove anything that is co-bound but weakly bound. And then you can either elute, add some reagent that will release your captured target, and then do downstream analysis of this. Or you can also, in very many cases, just do the analysis while your capture target is on the bead. So you can get a very sensitive detection or manipulation of the molecule that you have bound to the bead.
Nice. It sounds like a very simple, straightforward technique. So interesting. And is there anything else around? Like, for instance, I perhaps have many samples that I want to process. Have you got any tips around how I can still use Dynabeads if I have many samples?
Yeah, well, Dynabeads is especially nice if you have many samples. So because of the ease of automation and the amount of new equipment for handling magnetic beads in automated procedure that has been developed over the years. So Thermo Fisher has their list of menu of instruments, but also any laboratory pipetting station can be adapted to be able to work with magnetic beads in high throughput manner. And also when you produce these beads with the fish hook, that can be done in really large scale and can be quality controlled in large scale, and then you can distribute this into all your hundreds or thousands of samples. So it's also in many cases a very rational and cost-effective way of doing large scale screening and testing.
Sounds incredibly scalable and versatile, and a simple process made even simpler by automation as well. So
so automation is a key feature you should always consider when working with Dynabeads if you have more than a handful of samples.
Yeah, it's good to have that scalability option as well. Very exciting. So we've learned a little bit about some of the applications. Maybe we know how they work now. How can we apply them? What areas could we apply Dynabeads to?
Yes, and this is some of the fascinating things and why I've been enjoying this long period working with Dynabeads, that we always see new applications coming up, even if this technology is well established and not very novel. We still see that there are new techniques that can use this technology, putting new challenges on how we make this and our quality, etc., but that it's still in development. And now maybe the areas we are most focusing on is for multiomics, where researchers want to really not only look at one information layer, for instance proteins or nucleic acid, but really want to combine all the different layers of information that is in biologics and also in the field of more towards liquid biopsy applications where you have you want to isolate different kinds of targets from your sample. So,
very good. So it seems like these Dynabeads have definitely been used for a very wider range of applications. Are there any kind of numbers you can give us, any kind of like key performance indicators just to kind of indicate the level of impact that these Dynabeads have been having on the world in the last many decades that they've been around?
Yeah, I don't know how many publications that you can find. That I heard the number around 70,000 different publications.
Wow.
We also tracked Nobel laureates within medicine and biology, and counted how many of those researchers have used Dynabeads, and that was also quite impressive. So it is really widespread within the community and a standard tool that is used in all fields of natural sciences and medical sciences research.
Incredible. So we are coming to an end, but thanks again for taking us on this incredible journey to learn more about the origin of Dynabeads and how they're really being applied. It's been a real great honor and a pleasure that you could join us today. But before we round off, just one last thing to ask you: what's next for Dynabeads?
Yeah. No, I mean, we have touched upon these next technology developments within multiomics. There we will see more and more use with miniaturizations. So you could think normally if you see Dynabeads in large tubes like this, but we also have a lot of new research and publications showing that you can use also Dynabeads down into nanoliter microchips, microfluidics technology. So there we will see an explosion in the different applications. So yeah.
More to come.
Definitely more to come, and definitely more to learn about more details on the different applications here, because this has been a nice conversation on high level.
Yeah, we can dig in deeper on some future episodes, hopefully. So hopefully our listeners can join us for that. But for now, thank you again for taking us on this journey, and thanks to you and all your colleagues for the incredible work that you do with the Dynabeads, for all the researchers and the patients around the world. For sure, Dynabeads is having a big impact, and so are all of you too. So thank you again for joining us.
Oh, thank you. Thank you for listening.
Cut.